Today's February 18th, 2021. I'm BrentNally, and I'm really excited to be here with a PhD, Dr. Rodolfo Goya. Dr. Goya has a PhD in biochemistry, clinical biochemistry from National University of La Plata in La Plata City, Argentina. And Rudy is what Dr. Goya likes to go by. So Rudy has been interested in slowing, stopping, and reversing human aging since a very young age. And I'm going to kick it to you, Rudy. Why don't you share with us a little bit of your background, your interest in aging, what kind of research you are conducting at this time, and then we'll get into how to maybe solve the human aging problem towards the end of this interview. The floor is yours.
Okay, thank you. Well, first, let's start to. I will say again what I was saying before the recording began, okay? Because I think that, as you said, was good stuff. Well, I am quite confident. I am a non-believer, but I am a strong, confident person in the human species because of the achievements that we have attained in just a few thousand years, which is a blink of an eye in a cosmos time. So in very few millennia, we just dominated the planet. And so we have, and we always do things in the end that Mother Nature apparently doesn't want us to do. For example, we shouldn't be able to fly like a bird, and we fly better than birds. We are not supposed to dive deep into the oceans, and we do better than many other species, and so on. So we are continuously breaking the laws of nature. But of course, this is a sort of an image, because really nature is not putting constraints on us. It's just we are evolving. But what I was saying to you before is that anyway, if I am sure that we will be achieved the cure for aging, for biological aging, but we are facing now, even if I was 25-year-olds, limitations compared to the technology we have created, and we keep creating it, perfecting more and more. And at some time, that technology that is in many cases above us will begin to design their own avatars. And those avatars, of course, will be created by technologies, by beings that will be superior to us. So we will be pressed not to be left behind as a primate species, to merge, as Ray Kurzweil anticipate. I agree completely with him. With our technology to become also avatars, because otherwise, in this world where information is exploding and nobody can cope, Leonardo da Vinci was able to know everything, anatomy, painting, of course, everything. Today, nobody can even know his or her own specialty, because everything is growing so we are becoming very limited, even if we are very young. That this is not a problem of aging, it's a problem of our limitation, our carbon-based biology limitation. So in the end, perhaps we can go through a transient time where biological aging is defeated. We can continue to be young indefinitely. That would definitely be. But even in this situation, we still be needing something else to be enhanced, because we cannot coexist with our technology, our avatar, which will be more intelligent than us. So we will want to become one of them without losing our identity. That is very important. Without losing our identity. Well, these are just philosophical remarks. So in the end, I think that we will have to merge with our technology. Defeating aging is not enough now at this stage of evolution. We need to improve ourselves, and this will take thousands of years or more in terms of evolution. Evolution works very slowly, and technology works very fast, exponentially. So we need to just use technology to merge with our technology. After all, technology is our child, so we should have the right to do it. So we will evolve in a different way, but it's part of evolution. So again, we will be breaking a law that Mother Nature that says that we should perhaps. Well, this is what we think. It says evolving biologically. This is no longer possible. Okay, but this is for the future. Nevertheless, now this is what I can foresee. When I began to be concerned about or interested in the problem of aging, I I am an odd person because when I was a teenager, uh, I just observed when playing with my friends, uh, little animals, insects, living things, and for some reason that I don't understand, those really impressed me vividly and marveled me because they were living things that were I realized that were quite complex. So the death of one of those very complex and marvelous living things, be it an insect or a small animal, well, was such a loss of information and of complexity, loss of something very so beautiful and complex that it seemed that the destruction of life was really a sort of waste or something very beautiful. Of course, I didn't contemplate turnover, survival of a species. I was just a teenager. And then I began to realize that, well, everybody ages. This is a fate that Mother Nature has imposed to every living being. And this will include, I was 13, so the future lay far, far, far away from me. But I was a little bit more concerned about my parents that were young at that time and my grandparents that were not so young. So I realized that I was going to lose them, these beloved persons, in, in a future was not so far away. And that makes me feel as a sentiment of rebellion. I rebelled at this fate, universal fate, that Mother Nature was imposing on all of us, and that we didn't want to follow. I didn't want to die. Apparently, nobody wants to die. Not even that small insect. So I thought, then I was in my 17s, that such a problem, such a transcendental problem, deserves that a person devotes his or her whole professional career to solving it. That was a problem transcendental enough to be able to just um, take your whole career and effort trying to solve it. So I decided at the time, my knowledge was very, very shallow. Uh, I was just finishing secondary school and had to enroll, had to choose a university here at the city of La Plata. I was living in a different city when I was a child. So I could choose, according to my information, medicine, because of course, traditionally, if you want to defeat aging, seems that medicine could be. But I have the feeling, without much information or knowledge, I didn't have any relative being biochemist or scientist or something like this, that the secret of life, the inner workings of life, should be in the molecules, in the chemistry. Well, don't ask me why, because I didn't have really much information, just gut feeling. So definitely I said, no, I will enroll myself in biochemistry, which was a career there, clinical biochemistry. So I enrolled in that, and with the years, I realized, was just pure good luck, that I made the right choice, because in fact the secret of life, of the more intimate mechanism of life, lie in biochemistry molecules. Of course, it's more complex than that, but really you have to study molecules, not really whole body, to be able to reach to those fundamental mechanisms that, well, drive our aging process. At the time, it didn't really occur to me that rejuvenation would be possible. I thought that it would be possible. Then that was just going forward when I was at the university, was getting more information. Um, so I thought that perhaps that is beyond my time. Nanotechnology possibly will be Drexler. I read Drexler books and I was very impressed, and I thought that's the way. But this is a very different quantum jump on science and technology compared to what we have had at that time in the, say, in the 60s or early 70s. So, but I really didn't dream of being able to see biological rejuvenation being achieved during my lifetime. So when I began, then, well, I got my PhD degree. Then I was awarded at NIH from the Fogger International Center to spend 3 years in Michigan State University working with a pioneer in neuroendocrinology of aging, that is the brain and the hormones. There is a master gland, the pituitary gland, that hangs just from the brain, from the bottom of our brain. And that everything is controlled by this little portion, region of the brain known as the hypothalamus. So the hypothalamus controls the pituitary gland, and the pituitary gland, being a master gland, releases hormones that controls other glands of our body, thyroid, you name it, adrenals, gonads, virtually all of them. So this is a hierarchical organization that happens in living beings. So this is not just for humans or for mammals. It's general. It's a way life organizes itself. So we have to try to think like nature thinks, to try to understand the mechanism that we are trying to explore and to harness, perhaps for our benefit. I studied the aging of the neuroendocrine system. That was the 70s. And before that, it was not possible. There was a lot of interest in it had been discovered that the hypothalamus had a connection with the pituitary gland, that all those things were being discovered there. So people realized that the neuroendocrine system, as it is called, the combination of the brain through the hypothalamus and the pituitary glands that allow us to be controlled in the processes that are not very fast. For the processes that are very fast, for example, regulating heart rate, respiratory rate, and so on, we need a system, a faster system, wires. Those are the nerves, sympathetic nervous system and the parasympathetic. So different nerves that just regulate things that need to be regulated very fast. If your heart is beating too fast, you cannot rely on the slow process of hormone release. There are some hormones released, but you need some mechanism to control very fast. So those are the nerves. So this is a combination of slow and fast control system. All of that is the relate point to integrate all that information is the hypothalamus. So it's a very important, it's very ancestral part of our brain. So I began to study that, but I was saying at the time, the problem was that there was not a method, reliable method, to measure hormones in blood, pituitary hormones, for example, because hormones are circulating in blood, in serum, in very, very, very tiny amount. So, well, it's, for example, most of the pituitary hormones are present in concentration expressed in nanograms per ml, which a nanogram is a billionth of a gram. So it's there, what is so tiny, and the molecules are relatively tiny, that normal protein detection methods are not sensitive enough. So in the 70s and the late 60s, a method was discovered by Gerson and Gallo that were awarded the Nobel Prize for that, to measure hormones by a new method called radioimmunoassay. The method used radioactively marked or labeled molecules, hormone. Well, that allowed, provided a tool to endocrinologists to be able to, for the first time, measure hormones. And so study the changes that happen in the neuroendocrine system with aging, because it was obvious for everyone that being so important, the neuroendocrine system and the hormone, to control different processes, puberty, for instance, puberty is triggered because the hypothalamus ordered to start releasing certain hormone, and so puberty comes. So this is a program process, of course. Well, people began to do studies. At that time, the more popular laboratory animal was the rat, was not the mice. The popularity of mice came later because rats were becoming very expensive to keep, the 10 times larger than mice. So this kind of pressures pushed researchers to mice. But there is a lot of information from the 60s, 70s on rats. For example, neuroscience has a lot of information coming originally from rats. And I worked most of my life, still do, with rats rather than mice, because working with rats has some advantages. And in Argentina, it's not as expensive as in the northern hemisphere to work with animals, because there are problems that researchers work in the north that we don't have. So we exploit that comparative advantage.
And Rudy, we'll get into Didier Cornell's $25,000 donation and your current research on the rats with the young plasma transfusion. Just want to let the audience know we'll get into that later, because Didier was my last interview on this channel. So if people had watched that interview, then they got a little taste of what you're doing. So we want to get into those details later. So I apologize. Continue, please.
Yeah, I am one there. But we need a little bit of background. But even many researchers do not study too much classical physiology. So the hormone levels are changing when we age. Most of the hormones, pituitary hormones, go down with aging. So people discovered those facts at that time and say, oh, okay, so there is a decline in growth hormone levels in the blood, in the old people and old animals. Thyroid hormone declines also, and so on. So here's the solution. Now, we have just to give hormone replacement therapy to those old animals and eventually people to replace the hormones, because they are getting low because of the aging process in those hormones. So I was involved in this kind of hormone replacement therapy experiment. And I remember one study we did in our rats, injecting every day growth hormone and thyroxine intramuscularly to all rats. So, and it was striking to us that at the beginning, for 1 week or so, the animals really changed, were revitalized, because I remember the fur of the animals became much healthier, and the bright of the eyes also was something that you could see. So that was, I thought, okay, so we were right. They needed hormone replacement. That was the solution, hormone. But 10 days later, the animals become become more refractory to the treatment. We needed to increase the dose because the same dose would not work. And so in a short time, the animals stopped responding to the treatment, even if we increase it. So that didn't seem to be the final secret, because what happened? The cells in the target organ have receptors, and those receptors can be downregulated. So if you provide too much of the hormone, they are downregulated and the cells become refractory. So in the end, hormone replacement was not the way to replace. If I remember the hormones and other molecules, just the transit system in the body is the blood. The blood is not evolved to give orders. The organ that is evolved to give orders is the brain, and eventually the endocrine system. This is just evolutionary purposes. So sometimes people say, well, everything is in the blood. The blood is controlling everything because young blood rejuvenates. And so the blood is the secret. No, the blood is just a transit network. So if you are looking for the source of rejuvenating molecules, you are looking at the wrong place. You are just using plasma from, like we are doing now, from very young rats. I inject them into old rats. And of course, those old rats are receiving higher levels of hormones and other factors because they are coming from young animals. But this is just like giving a drug, perhaps. So what Didier says, and this is, I fully agree with him, is that you have to provide, it's okay, you do an experiment like the Conboys do and prove that, I don't know, using plasma or whatever component of blood from young animals rejuvenates all that. But you have to do this, so it improves cognitive function, memory, so on. So people say, okay, the conclusion is that young plasma is a rejuvenating substance. Okay, but if it is really working and extending life, doing experiments where, like the one that we are doing now, administer your treatment, whatever it is, to your rats, old rats, not to have to wait too long, and see if they live longer than your controls. If they live longer, nobody will really question you or have any doubt that your treatment worked, because to extend the life of the whole animals, you need that everything has been improved in your body. So if I say, Brent, I treated just as a fake, of course, scenario. I treated a person with aspirin plus something else, and every month I injected in, or he took an aspirin with the other component. And this person lived, that was just one patient, until 185 years. And so what is the end value of the person of your experiment, Rudy? N is equal to. So you may say, well, you are not going to publish that because an end value of 1, the reviewers will laugh at your study. Yes, I say, that's I understand. But show me humans that are 185 years old. So it cannot be chance that I picked up that person as a volunteer and happened to be a person that naturally lived so long. So you have to accept that something, the treatment that I provided was working. I mentioned this because we had an experiment with a single rat that was my more successful experiment that lived treated with adult stem cells, human stem cells. And that rat was a single rat, lived up to 44 months. The maximum lifespan of rats are the average albino rats, white rats, 3 years. And this rat lived, the natural lifespan was 8 months beyond that. That was, I surprised me because I didn't expect. So we published that because it was just.
And Rudy, that's about the longest a rat has ever lived from my research, about 3.8 years. Is that right?
As far as I know, yes, of course. I showed the data to Aubrey the Great. And Aubrey, who is a very perceptive man, said, well, you are sure, because that's the first question, are you sure, Rudy, that there is no confusion, that this is not, it was impossible because I couldn't just go to my animal rooms and pick up a fake rat that is 44 months old, because those rats do not exist. I mean, if I show you a man and say, this person is 185 years old, it's difficult to say, are you sure that this is your volunteer? Maybe that you pick up another one or just fake it. It is impossible to fake that because there are no persons 185 years old. This is the reason. Well, that I try to repeat this, but those experiments take, because the treatment began when the rat was young adult, 5 years. It's a lot of time, a lot of money. With one rat, no problem. But if you undertake such an experiment with, say, 10 rats or 20 rats, which is the right thing to do, that is, they will take 5 years, a lot of resources, very labor intensive, and it will just use up, you may burn the career of a PhD student because if things do not work as they should. So this is why people are realizing of this possibility, but nobody does this kind of experiment because it takes a lot of time, 5 years, and especially if you work in the States or Europe, where keeping animals, especially old animals, is cost prohibitive. It's very, very expensive. Here it's less expensive, much less expensive, but still it takes time. So this is why I didn't repeat that. But I tried the same studies. I mentioned this because it's quite important in older rats. The rats, the age I am working with right now, today, and the results were much weaker. So when I treated the animal being already old, the power of those improvements was much, much weaker. So definitely, I realized that I have to start when the animal is a young adult. Why? Because stem cells, human adult stem cells, are not rejuvenating the rat. What they are doing is slowing down the aging process, the rate of the aging process, because they somehow stabilize homeostasis, the whole regulatory system of the rat. So you have to regard animals like complex totals, not just the blood or cells. Well, but I hope that someone with the resources to do this would repeat the experiment. This is why I wanted to publish them. Aubrey shared my view. That should be known because it was something important. And this is, everybody can repeat this. Adult human adult stem cells are available for everyone. Old rats are available for everyone. So this is nothing, no secret. You can just go to your lab and repeat that experiment. I am positive that you will get the same results, especially because I was skeptical at the beginning. It was a friend, the first author of the paper, Dr. Mancilla, that convinced me because I didn't believe that stem cells would extend life. So I reluctantly accepted to give him one rat because he was convinced. And he was right, and I was wrong. I am glad that I say yes at the time. Well, that is a story and that approach to our system. So,
Rudy, can I pause right there? I want to fill in a couple of very quick gaps that the audience might have. A few minutes ago, you mentioned the Conboys. So that's Michael and Irina Conboy. They're at University of California, Berkeley. And they've been around for decades, but the new research that they've done that people are excited about is their plasma dilution of mice and showing age reversal from those mice. And so what you're doing with the rats is you're doing the young plasma transfusion. And so the Conboys, are basically saying you don't need to do the plasma transfusion, plasma dilution is enough. And so you, I believe, disagree. We had the HEALS conference on February 11th that Didier Cornell hosted, and we had lots of fantastic scientists there. You presented, Irina and Michael Conboy presented. Many other people presented their research. Harold Katcher presented. Hopefully I'll interview Harold soon. He's, I think, just having issues with internet or busy. So I'm trying to get that interview with him. So all this kind of ties together due to Didier Cornell's $25,000 donation to Harold so that his team can do maximum. And then yours as well.
One word about the Conboys. Yes, I had a chance at that meeting to discuss things with Michael and with Irina Conboy. And I asked them, do you think that it would be interesting to do an experiment like the ones that Didier is just supporting for Harold and for me, just to see one of your treatments, the plasma dilution treatment, can get mice, relatively old mice, not to have to wait too long, to live longer than control animals, because that would be definite proof that you are rejuvenating. Because improving some parameters, only that, is not enough. If you say, well, the memory improve of the animal, that's a short experiment that ends. So did the treatment really prolong the lifespan? Well, you have to do the experiment. Of course, beginning with young mice or rats is difficult because it takes 5 years. So nobody will be prepared to spend 5 years on an experiment. But if you are treating old mice or rats, as Harold did, when they are old already, they have a few months to live. And like in the Harold's case, they seem to be rejuvenated. That's something different, because this is why, well, Didier doesn't believe much in that. I am quite cautious also. So he wanted to just spend money just to make sure with a conclusive study that let's focus on elixir now. Elixir was able to not rejuvenate, but at least extend life. And let me discuss, because I was involved in the study. In fact, my contribution, the contribution of my lab, was not very important. In fact, the star is really hard. I was trying to convince Steve Horvath for some time, and before that experiment was done, to set up an epigenetic clock for rats, because I was working with rats, and everybody was focused on mice and humans and maybe perhaps dogs. So I said, Steve, I have a colony in Argentina of rats, female rats, of different ages, and they are at your disposal if you would like to set up a clock for rats. He said, well, that would be interesting. Yes, could be. And then he met, I didn't know that at the time, Harold. And Harold came up to him with this experiment. So he said, okay, maybe the right time to set up a clock. So one day he invited me to participate and say, could you provide me with some of your rats? And actually, I provided him with 70% of my colony at the time. I committed all my animals because I was dying for having the epigenetic.
Steve Horvath. He provided Steve Horvath.
Steve Horvath, yes, exactly. Right. Different organs and brain regions, this is an advantage of the rat, is large enough that allows you to collect brain regions like the hippocampus, the substantia nigra, which is important in Parkinson, and other regions which are important. So you can analyze the epigenetic age of these regions, because epigenetically speaking, they don't necessarily age at the same rate. Okay, so you can measure that if you have the different regions and extract the DNA. So he studied also, well, heart, skin, and so on, blood, of course. So I provided this base of rats to be able to set up the clock, the rat clock. Then, when this clock set up, he could measure samples in Harold's animals. So Harold sent him blood, liver, heart muscle, and hypothalamus. It is that important part of the rat. So he ran on those, he used 3 groups. Young intact animals, 3 months after the indoor study, 25-month-old males, male rats controlled, and a third group of 25-month-old rats that he treated for 5 months with elixir, which is a plasma fraction that he doesn't reveal the composition, but he prepared, and he has been experimenting with that for years. Okay, he won't tell, or let me have an aliquot of that, but this is what he uses. And when Steve measured those samples, did the DNA determination, and calculated the epigenetic age, something really quite unexpected and remarkable occurred. The epigenetic age of those animals, I refer to liver, heart, and
hypothalamus.
Hypothalamus, and there is another thing that I am forgetting. Okay, liver, heart, and something else, and the hypothalamus. The hypothalamus didn't rejuvenate too much, but the other 3 organs, I am forgetting one now.
Kidney, really, wasn't it the kidney was the last one?
Yes, kidney, exactly. So these animals showed these organs an epigenetic age from the treated rat that was close to the young animals because they used young controls. At the time, those animals were 7 months old. So this epigenetic age of the old treated rat, which chronologically were 25, 26 months old, but epigenetically those rats, those organs, were like 7 or 8 months old. So there was remarkable and unprecedented epigenetic rejuvenation. So the epigenetic clock was set back several months, that in the life of a rat is a lot. So taken from, say, 70 years old in human scale to, I don't know, 25 years of age for a human. That was really, and Steve said, well, this seems to be too good to be true. And at the same time, they measure in those animals a number of parameters like hematological parameters, cholesterol and other things, cytokines, and also some functional parameters, particularly grip strength, the muscular force of the animal had, and all of those apparently had been improved significantly. So were the animals rejuvenated? Because there is an hypothesis, just some people play with that, including myself, that since there is such a good correlation with the epigenetic age, that is the profile, the methylation profile of the DNA in humans, but in other animals too, that perhaps if you accelerate biological age, the epigenetic clock will tick faster, and so on. And in centenarians, which live a longer life, the epigenetic clock ticks more slowly. So because we have like a number of tickings of the clock during our life. Hummingbird have the same number of ticks allotted like you and me, or than an elephant. But he uses his epigenetic clock ticks very fast. So he uses that number of allotted ticks in maybe 3 months. And the elephant just ticks very slowly. It's like the heart, right? And so it takes many, many more years. And the same happens with that. So,
Rudy, real quick, since you mentioned elephants, the interesting thing is they can actually live a lot longer than Mother Nature allows because they have 8 sets of teeth, and they come in when they lose their 8 sets of teeth. If they don't have help from us humans, they die because they can't chew their food and they die. But I visited an elephant park in Thailand in 2018 with my wife, and we learned a lot. We walked around and saw like 90 elephants or something, and some of them were over 100 years old. You can tell because their ears never stop growing. So the point is that we can extend life even further and further and further with all these different animals, the more that we understand about how they age and how we can improve their life. So just a little fun segue about elephants.
No, because that has a limiting factor for the life of the elephant. If you correct that factor and the animals can keep feeding themselves, they will live longer. Up to a certain point, probably you won't get an animal that whatever your intervention will live up to a millennium, just to exaggerate. And we can extend our lives and live up to 120 with a good quality of life, but we never live 200 years, probably because we're not made to live so longer. Same thing for a rat. A rat lives 3 years, and by 3 years, it just suspect. Maybe it may live 3 years and a half, but will never live 10 years. And this is something to discuss, but I don't want to discuss this because I will go out of the focus. So when I saw, because I have spent my life handling rats and animals, when I saw the videos and the data from Harold, I realized one of the controls they did was to weight the animals, just the body weight of the animals at the beginning. And in the case of male rats, they grow very big. So when they are young, they may weigh in grams 350 grams, more or less, which is about a pound, a little bit. But when they are old, they reach half a kilo. I think it's that close to a pound. Well, so like in human happen, they gain weight. When they weighed the treated old rats and the control old rats, the weight hadn't changed. It was the same. So elixir didn't have any effect on the body weight of the animal. And if you are rejuvenated to an age to 25 years of age, I am sure your body weight was a little less at that time. So, but you say, okay, uh, Brent, your epigenetic age, what says to you, has to rejuvenate, but you feel that your body is the same. So that's good news, but it's theoretical news because you feel like having the biological age you have. Well, this is what happened with Harold's rod. So they were improved. So say partially rejuvenated, but they did not rejuvenate. So it's a hypothesis that
Rudy, what's your, I think you're getting to it right there. What's your hypothesis on why they didn't rejuvenate from
well, at first I had the hypothesis that the epigenetic clock, that methylation profile, since they are part of the epigenome and these are at the heart of the command center of the body, which is DNA and chromatin. Chromatin is DNA plus the proteins, the different proteins that wrap it. So this complex is called chromatin. That's quite important. Perhaps that was part. It was not just a biomarker. Perhaps it was part of the driving component of the epigenome, the driving component concerning aging, because it is quite clear now from the cell experiments that the epigenome and not DNA is the driver of aging. And this is why you can reverse aging, because the epigenome allows you through the Yamanaka gene to do that, whether there is no, if with the cumulative theory of aging, you have no chance, it's not contemplated that aging is reversible at all. You can slow it down, but never send back. The epigenetic model of aging says that, and it has been done. Rejuvenation has been achieved. It's not just a theory. Okay, so perhaps that was what the LKC was not only a marker but something else. But this experiment that revealed to me that definitely the rats were not being rejuvenated, at least not to the same extent that the epigenetic clock, revealed to me that well, at least the clock may be part of the driver, but it's not all of it, because the clock was going in one direction and the rat kept aging. Was essentially old, improved maybe, but old. So Didier came with this proposal, and this proposal was wonderful, and it was wonderful that Harold accepted to do this experiment, because now we will know. Let's imagine two scenarios. The experiment is underway now, so he is administering elixir to the old treated rats and nothing to the other rats, to the old control. So these rats are 25, 26 months old, perhaps now, and this year the controls will die, because we not go past this year, because in several months they will be quite old and they will probably die, because it's just what happened. And then we will see if the treated animals with elixir live much longer than those controls. So let's assume that the controls die when they are 33 months old. Okay, this is what will happen here. The other animals, the treated animals, should live at least until next year for you to be able to state that they significantly, markedly extended age. Remember my experiment with my old rat. So at least to, I don't know, 38 months or something like this, a difference sufficiently big with the controls that you say, okay, elixir doesn't rejuvenate, because rejuvenation will not happen. That I can anticipate that. But it's a powerful life extending preparation. Well, that's good, because I have nothing against the life extension. I would be happy. So, Harold, congratulations, you have discovered a powerful life extension preparation. The problem is that if at the end of the day, Harold finds, and this is the time of truth, that both groups live more or less the same. So that ends everything. That is will be a bad news for Harold, will be a bad news for Steve, because now
It'll be bad news for all of us. Bad news for humanity.
Yeah, for humanity, because I really very much like that Harold is right, because having elixir with the properties that apparently it may have will solve a lot of problems. Doing rejuvenation experiment with the Yamanaka genes with the transgenic animals is not easy. It's quite challenging. What can be done, but if you have a transgenic animal and you express the Yamanaka genes for too long time or too strongly, you get tumors and the animal die. And if you express the genes too weakly or for too short of a time, there is no effect. So you have to just work on a tight rope with that. With elixir, you have no such effects if it works, because you just get rejuvenation, probably some partial reprogramming, we don't know, but you will have an effect on epigenetic age, but also this is what is important, on the phenotype, because what it matters is the phenotype. If you tell me, good news, Rudy, Steve Horvath measure your blood epigenetic age, and you are 25. Oh, wonderful, Brent, what a good news. How do you feel of 69? Okay, so I would prefer to say that I feel like a 25, and you say, well, sorry, Rudy, your epigenetic age is still 69. I will be delirious because I will feel like 25. So that's the point, is rejuvenate really. But even if the epigenetic clock is just a marker, a very good marker of age, it's a quite useful tool. That is important. But we will know that it's only a marker. It's not involved in the in the power of the epigenome to drive aging. There is no doubt that this is the case, because the experiments that were conclusive were the experiments with cells that are not too much discussed. These were a complete success. For example, there was a French group took skin fibroblasts from centenarian, healthy centenarian, cultured them, reprogrammed them to iPS, and they rejuvenated completely. And those cells, when were cultured initially, they were showing different telomeres were shorter, mitochondria was not working well, not because the patient was diseased, but because he was old. And so there are many dysfunctional features of those cells. When he got the cell to be iPS, all that disappeared. The cell was young, but of course lost its identity. That's the main problem. But they were cells. So they differentiated back the cells that could be done to fibroblasts, skin fibroblasts. And so they got back fibroblasts, like the original cell type, but they were rejuvenated, cannot be distinguished from a fibroblast taken from the skin of a teenager. So that was the alchemist dream come true. And this is an achievement, marvelous achievement. So rejuvenation has been achieved, and then the epigenome, the epigenetic age, which of course went to zero, and phenotype was working parallel. So it was a change in epigenetic age, which was paralleled by a change in phenotype. So there was a rejuvenation, and also, of course, epigenetic rejuvenation. This is not the case in vivo so far, because of course in vivo is a different thing. Working with cells is like one thing. Working in vivo is like adding a third dimension to the problem, and that things become more complex. And I realize now, I was very optimistic after the publication in 2016 of Piso Belmonte, that I said, well, that would enough work be achieved. But now I am realizing that it's not so easy to do that. So I am a little bit more cautious. So this is the idea of the elixir study, and this is what the importance of the initiative that Didier has taken. In my case, I am just doing experiments with young plasma, which you can consider sort of a diluted elixir. So far, the survival curves don't say much, but we are going to let the animals live until the end of their life. And then perhaps there was some difference, because there's a long story, controversial sometimes. People saying that young blood is really, or young plasma has rejuvenating properties, life extending properties. Okay, let's see what happens when you begin the treatment in old animals. And in a few months, we will know. And we agree with Didier, I think the same agreement was made by Harold, that the data will be published whether they are negative or positive, which seems to be okay, because negative results are also informative for people. Just avoid them to do the same wrong experiment. That would be difficult for Harold. He's risking things, but you have to know what is the truth, what you're having in your hands, that it is really working. If it doesn't work, it will die. It will die soon, because other people will want to repeat independently that experiment. So take the example of Gordon. Other people could clone animals. This is why animal cloning became a fact. Yamanaka genes were used for many other people, and they could reprogram themselves, and so it was accepted as a fact. But if not, nobody can repeat your experiments. Very proud.
Yeah, definitely. And just a real quick plug on Yamanaka factors. We have a new YouTube channel where I now host Lifespan News. It's called Life Extend Show. I'll put a link in the description below. And on Monday, Nicola and Sadir El Shok and Nicola Bagala, they produced a video that came out on Monday, February 15th, about Yamanaka factors. So if you Rudy just gave a really good description of a lot about Yamanaka factors, but we have a short video in there that explains a few other things there. And yeah, it's,
can you send me the link to that? Because
I will definitely send you the link. It's a new YouTube channel that we have. We do on every Tuesday, Lifespan News comes out. I'm the host of that. It's 5 stories of recent, relevant news stories in life extension. And then every other Monday, we have science and advocacy videos, episodes about various life extension topics, like Yamanaka factors, for example. So I will definitely send you the link to that, and there'll be a link in the description below for everybody watching here on YouTube. And so, Rudy, one thing that I want to make sure the audience is clear on is the reason Didier gave this $25,000 donation to Harold's team and then to your team in Argentina is specifically to see if we can extend the maximum lifespan of these rats. And so why was that not done at the very beginning for Harold's group? And also, why are the Conboys, why are they not focused on that? That is really an important question that to me is just so obvious. We should be, I thought we were trying to extend lifespan and health span. I thought that was the whole point of kind of everything we're doing. So why would we murder these rats at a certain point in their life and not see how long they actually are living with these therapies?
Well, this should be done. If you ask me or someone else to do an experiment beginning with young rats, I will tell you, Brent, this will take 5 years. And this is a material consideration. And also some expenses. Even if I do that in Argentina, where my expenses with animal are low, these studies, if you use a sufficient number of rats, are labor intensive, so you have to prepare cells. Well, it depends what you are using. So now in these experiments we are involved in, well, they take some time for us because we have to take care of the animals, inject the animals every so on. So we take blood samples, and so we are studying cognitive spatial memory at the beginning of treatment and 3 months right now into the treatment, despite of the fact that we will let the animals live. But we want to know what happened in 3 months with the controls and the experimental animals, just to have some ideas. But at the end, they will die one by one, so we will not be able to kill the animals at the end because they will be dying progressively. This is another disadvantage of letting the animals live their natural life. So, okay, you have a very strong information, but you cannot collect data from the animals because they are dying one by one. So you have to set up, but the results are quite conclusive. If I tell you, Brent, my rats treated with young plasma live, say, 6 months. Just I am exaggerating, but let's assume 6 months, 6 months longer than the control. So you will definitely say me, okay, no question that young plasma is extending the lifespan of your old animal, even though the animals are really old. So I'm taking a deteriorated homeostasis. Things are not working like clockwork, like they were when the animals were 3 months, but still they could respond and be stabilized and live 6 months longer than the control. So that would be an achievement because, of course, using preventive treatment like the one I did to that rat, well, young people in practical terms will not like at 30 to begin to inject himself, most people at least, something just to stabilize their homeostasis and live longer because they say, well, this is colony, I feel perfectly well. I will wait until I am really a little bit old. But for a life extension product, I think that this is the best way to work, letting it work from the beginning. But if you have a rejuvenating stuff like elixir, maybe that is wonderful, but it rejuvenated apparently animals that were already old. It's like the Yamanaka genes in this sense. You apply the Yamanaka genes to cells like those from those centenarians that are very old. They are near the end of their life viability, and then they receive commands, signals, orders from some genes that Mother Nature developed. It was not a personal engineer, it was Mother Nature over hundreds of millions of years. So it's time proven. And those cells responded, and responded in nothing less than becoming rejuvenated. You have to rebuild the structure of your very deteriorated cells and become a brand new embryonic cell. That is a miracle. These four Yamanaka genes, this is what I am so impressed and I feel wonder at those genes because they do things by mechanisms that we have not the faintest idea, but they do, and could be used to rejuvenate because this is what they do. Well, they do two things. One that we want them to do and to use, which is the rejuvenation. But they do another thing that we don't want them to do is the differentiation. They just take away cells' identity. So we have to work on this genes trying to just use the good action and neutralize in some way the bad action. This is not trivial, but it's something that we have. And I am confident on these genes because it was nothing that was created by a person, engineer, and because if I have an artificial substance, life in a animal, everything is connected with everything, Brent. So if you touch something here in your body, you are affecting your brain, you are affecting your liver, because everything is connected with everything. But Mother Nature developed that over the years, and so we are sure that they shouldn't have side effects that we don't want to have, except the ones that we know already. So, but this is challenging. They will need more technology, more people working on that, and more funds, of course. So the studies that David Sinclair, for example, are doing, and Jesus Avila in Madrid, are very good because we need a critical mass of people beginning to work on this. This is challenging. This is not difficult. It's not easy work, but the results are quite significant. Breakthrough. The recent paper, probably you saw it, by David Sinclair in Nature published. They restore the vision of animals. I have a review. I think I don't send it to you. Just is now released, describing those experiments, discussing my interpretation of the experiments from David Sinclair. And he has also a hypothesis about why all cells respond to the Yamanaka gene. So I discussed that hypothesis And so on. I will send you a copy of that, because if you have the time to read, especially what the second part, which refers to cell rejuvenation, to me, cell rejuvenation is something fascinating.
It's extremely fascinating. And we did do a story on Lifespan News about the work that you're referring to there at Harvard Medical School with Dr. David Sinclair's lab. Very fascinating stuff. And it's actually getting mainstream press. Just a few days ago, Bloomberg did a story on uh, yeah, it was Bloomberg did a story, and it got a lot of views on YouTube that I saw, and that was just hours after it came out. So I'll check that video. We're talking maybe hundreds of thousands of views. So this is going more mainstream to a certain extent with things like that, which is good. Rudy, I want to kind of go more rapid fire with a few things. I have a list of questions here. So first of all, the rats are Sprague Dawley rats that you're using in your lab and that Harold's using. Is that correct? You're nodding. Yeah. Okay.
They are female. Mine are females. His are males.
Okay, great. Because we know that the reason that's important because there's different species of rats and we know they have slightly different lifespans and things. So just wanted to clarify that. And then Harold believes that the circadian clock is a timer for our lifespan. Do you share that view with Harold?
No, I don't. I have, as a endocrinologist, studied the circadian clock, the pineal gland, and so. So this timekeeper to synchronize animals with day, the light of the day and night. And so this is mostly what I think they do. I may be wrong, but I don't have a gut feeling, which I work very much with gut feeling, that the circadian clocks are involved in are just part of the pacemaker of aging. Aging has a pacemaker, which of course should be in the epigenetic of some control cells, some master cells in the nucleus. Where? Where else than in the brain? Some place of the brain. A candidate region is the hypothalamus. Well, the circadian clock has a center, center of control, which is in the optic chiasm, which is in part of the hypothalamus. So all these vegetative control centers are there. So I would look for the center that controls the epigenetic clock there. But anyway, I have no evidence, nothing that inclines me to think that the circadian clocks. Of course, there is a hierarchy. So cells have their own intrinsic cycling, and which is interesting, Brent. I would like to mention this. You have an epigenetic clock, and the clock ticks. Okay, so and you have also cells have an circadian clock that also ticks intrinsically. So, but when this happens, after the cells, the young cells, the embryonic stem cells, begin to differentiate. While they keep as embryonic stem cells, which are just undifferentiated, they remain eternally young. Like you can keep cells for 1 year, and they will keep young. And in those cells, the epigenetic clock doesn't tick because aging is not, biological aging is not happening. And the circadian clock doesn't oscillate. They're wonderful. But the minute they differentiate, they begin to age, and both clocks begin to work. But if you reprogram the cells, say somatic cells, to IPS, then the epigenetic clock stops ticking, and the circadian clock stops oscillating. That is wonderful. So these embryonic cells are like in suspension, in sort of suspended animation. This is so biological speaking is fascinating, really. So they are pure youth, so to speak.
Yeah, that's super fascinating. Thanks for sharing those details there. I want to just quickly point out, I want to plug a different YouTube channel that I'm not affiliated with in any way. It's Modern Healthspan, and a gentleman named Richard runs this channel. And he did a series of interviews with Harold recently. I'll have a link in the description below. I only mention it because, I think that Harold ties in obviously to this discussion that we're having here. And if people wanted more information from Harold, they can watch those recent interviews of Harold Katcher done on Modern Healthspan. And hopefully,
I had
whenever Harold's available, I'll interview him.
I had an interview with Richard yesterday
Oh, great! So
morning.
you'll be on Modern
We,
Healthspan as well.
we discussed. We spent 1 hour and a half because the talk. I was monopolizing the talk. I am afraid, but it was quite. He didn't interrupt me, so I think that probably was not saying nonsense or rambling too much because I tend to ramble. But yeah, it was quite interesting. I really like very much to talk about my topics, and of course, my topic now, my final love is rejuvenation. Because before that, before all this happened, which is relatively recent, I was doing gene therapy in the aging brain, trying to restore, to prolong the life. When I discovered rejuvenation, aging, I lost interest in aging, because it's much better to study something that is just rejuvenating that a process that will end in death. So it's much more interesting to me now study and try to do modest things on rejuvenation than just studying aging. So I lost interest in aging, and I am really focused on rejuvenation. And I think because the best way to age, Brent, is not to age.
Yeah.
And for oldies like me, well, if you say, Rudy, you want to stop at 69? Yeah, please, Brent, help me. And if they offer me, well, stop at 69 and then go a little bit backwards to 50, I would say, I will beg you to do that.
Right.
It would be quite different thing.
And that's kind of why, Rudy, our tagline for our YouTube channel we told you about, our new one called Life Extend Show. So it's Life X 10 TEN show, Life Extend Show. So that's a little double entendre. But then our tagline is aging is getting old. And I didn't come up with that, but I think it's kind of catchy. And that's what we're trying to do. We're trying to make aging in humans old news by solving the human aging problem.
Yeah.
So I want to ask you just real briefly about Dobri Kiprov. I wasn't aware of him until recently. I had his private Zoom call with him a couple weeks ago. Maybe I'll interview him soon. Are you aware of Dobri's work?
No, exactly. Brief me on that,
So
because perhaps I know, but I am not relating the name.
Dobri, I found out about him from Irina and Michael Conboy's work. He's worked with them. He's in San Francisco, so he's geographically very close to, just across the San Francisco Bay. They're in Berkeley on the other side of the bay, the Conboys. And Dobri is doing human clinical trials with the plasma dilution type treatment, very similar to what the Conboys were doing in mice. However, Dobri has kind of he's keeping it secret what exactly he's putting in. With the Conboys, they use salt water and then they use albumin. With Dobri, he's using something that's proprietary. So I'm not sure. It could be salt water. It could be salt water plus something else. But then he is also putting 5% albumin back into the people. And just from the anecdotal, this is anecdotal, but there's testimonies on Dobri's YouTube channel of older folks, probably somewhere around your age. We're not talking like, centenarians or people in their 90s, but people in their 60s, 70s, what they look like. And they're having very anecdotal positive results. So they're already doing, similar type plasma dilution in humans. And I wasn't aware of that until just a few weeks ago. And I probably will interview Dobri soon to kind of learn more about that. So that's
Okay. So he's setting up a company, right?
Dobri is an MD and a PhD. He has a company. I believe he is doing clinical trials. He's doing 2 clinical trials, is my understanding. And I put links in the interview that I did with Didier on February 6, 2021, just a little under 2 weeks ago. I'll send you that interview. Rudy, in case you're interested in watching, because we talked about it a little bit, and then I put hyperlinks into the comments, the pinned comment in the description to kind of link to Dobri's video. But my understanding is Dobri's doing 2 clinical trials. One is an anti-aging clinical trial. So potentially, after running certain tests and diagnostics, somebody like me could potentially be a part of that clinical trial. Not saying that I will, I'm just saying potentially. But then he's doing another clinical trial on humans who have Alzheimer's to see if this can have any positive impacts on Alzheimer's as well. That's my understanding.
Well, if he has a significant restorative impact on Alzheimer patients, that would be an achievement. But in my view, this is my view, and may be wrong. I am really impressed by things that show categorically that work. Because remember, growth hormone, melatonin, and other, many people reported in the past that they were feeling well, better because of this treatment. And then at the end, you have to wait until time just convalidates that. So, and if you are running a company, say it's like George Washington was the manager of a cannon um, um, factory. He was doing a business and doing the revolution, the American Revolution. So if the British came and said, Mr. Washington, we offer such amount of money for all those guns, well, perhaps he could have decided to sell those to the British and to the American, because this is profit in the middle.
Yeah.
In cryonics, which is another thing that I involved in, foundations are not for profit. That is something that is important. Because if you are working on a product, whatever the product, and even elixir, you have to say that your product is good. Because if you do an experiment and your product doesn't work in that experiment, you will be reluctant to publish those. So if you have a company and sell something, this is my opinion, my personal opinion. You are limit. You are biased because of the result. So that's the point. You need the result to be good for your business. So this is why I am very bad. I am not gifted as a businessman, but I do not regret that, because with Didier, for example, he does this not for profit. So if the results are bad, he doesn't matter. He wants the truth. So do I. So he wants to publish them, and I agree, because people will know that this doesn't work and will perhaps stop, or have at least a strong proof that this doesn't work. If I was selling elixir, because there are millions, so it's difficult. I mean, I don't say that Harold would die, but we'll be in a difficult position. I am free of this difficulty. So this is why I think that science and the discoveries should be without looking for profit too soon. But this is just my view.
Rudy, that's a really important point, and I pretty much agree with everything you stated. So I just want to share a little bit. This is going to be like a quick little, plug, but I'm the co-founder and CEO of a new company called Longevity Plan. We're in very early beta, so we're not really promoting it a whole lot. But hey, I just said it on my YouTube channel. You guys can sign up, but we'll be improving a lot. But we're just trying to build kind of like, I don't want to be too grandiose, but kind of like the Amazon for longevity, in the sense where it's a place that we want it to be ease of use, good UI, good user interface, trustworthy. So we're 2 of our other co-founders are involved with Leaf. One is Keith Comito, the
I know him. Yeah.
And president of Leaf. Keith has become a dear friend of mine. He's an incredible person. He's a co-founder of our company, along with Paul Spiegel, who is on the board of Leaf and Humanity Plus. And so the point is we're not trying to actually do any of this research. So we don't have really, skin in the game or a dog in the fight of any of this research. We're trying to actually stay out of that so that we don't get any conflict of interest that you kind of just mentioned. But at the same time, we want to be very involved in reporting and have relationships with all this different, very fascinating research going on. So Better Humans, for example, is one of our partners. James Clements, 501.
I know him. He's a good friend of mine, James. We collaborate, in fact. Yeah.
Yeah, I've heard of.
Yeah.
I've heard of James many times over many years, but I hadn't checked out his work recently. And then last week, James and I we scheduled a 3-hour private Zoom call. We ended up talking for 3 hours. Very
Yeah.
impressed with James Clement. Go ahead with
We
anything you want to say.
we just published that review that I mentioned is very recent. I will send you a copy. And James is one of the authors because we were doing something in collaboration. Well, he's basically interested in supercentenarians and centenarians, but is interested in aging and in fighting aging. And of course, he has been captivated by rejuvenation. Perhaps I influenced him a little bit in that. He's one of the lines of research he has in his lab is on partial reprogramming on primary cell cultures to try to, well, rejuvenate cells in vitro initially. And I wanted to send, I have 4 students, PhD students, 2 of them to his lab to get some training, but I couldn't convince them because this is not a university. I said, it doesn't matter, he's very well equipped and he's doing it. We are doing it in my group, aging. So you should try. You are not going to spend 20 years with James. Just go spend 3 months and see what things are, because James is doing interesting things and you can learn there. But I couldn't convince because they think that they have to go to and stay at a university. For example, this is different ways to operate. I contacted David Sinclair to see if it was possible. One of my students was applying for a Fulbright fellowship, but then I learned that he had 30 persons working in his lab, and so he was looking for people that is productive. So a student is viewed as a manpower source, and this is not what I want my students to go there. Just that I want them to go there to learn things, to be productive, but not be just a pair of hands, nothing else. So I prefer smaller places, sometimes not so prestigious, but where my students can really learn more, receive closer attention. Probably to just as a student to have a conference and discussion with David, they would have to send him an email, and then in a couple of weeks, he may have the time to discuss things. And I prefer a more personal and closer relationship with the boss and the people in the lab. So, like relatively a small group. Well, she's now one of my students. Is going to work with Sebastiano, Vittorio Sebastiano, who was also one of the programmers of aging. So there are a few ones. Vittorio Sebastiano is one of them. David Sinclair is another one, a very important one, because he's a sort of agent of change, because that study that he published just gave visibility to this endeavor of this emerging view that the epigenome is really what matters in aging. And because, this DNA damage theory has been around for 50 years, and still you read a textbook and says that, and this is no longer accepted. Of course, it will take some time to be displaced, but the new data are coming very strong, and it's difficult to really challenge from the epigenetic side. So, well, this is times of change.
Well, Rudy, you teed me up there for kind of the big question that I like to ask a lot of longevity thought leaders, experts, scientists when I interview them. So the question is essentially, if you had unlimited resources, what do you think is the best way to ultimately solve the human aging problem to keep us young and healthy indefinitely? Or do you think that's not even possible no matter what we do? What is your thoughts?
No, I think it can be done. For science, nothing is impossible. This is one of my premises. Science and
I
technology
agree.
will, well, eventually, at the times of Christopher Columbus being able to speak with a cell phone from here to Sweden, would may, well, as Arthur C. Clarke says, any sufficiently advanced technology is indistinguishable from magic. And this is what technology becomes with time. And so now to keep a person living young for 2 centuries may look like magic. Not too distant not too far away in the future, this may become a routine thing to do because science and technology is moving at an exponential rate now. So everything is possible. So I think that will be achieved definitely. That was part of your question. Remind me of the rest of the question because I
Yeah,
responded.
the other part of the question is how do you think that we're going to do it? And what I didn't say that I'm now going to include is what's your timeline here realistically? Understanding these things are incredibly difficult.
No, I couldn't. I believe that Ray Kurzweil timeline is too optimistic. I may be wrong. But anyway, things will happen sooner that we think. I never expected to see biological rejuvenation being achieved in my lifetime. I'm just, it's happening in front of my eyes. So I am reluctant to just anticipate or to provide timelines. But I know that things will happen soon. But in the end, that is a first valid intermediate goal. In the end, Brent, we will have to merge with our technology. This is without, even if you and I were now 25, that is not enough to keep up with our technology. Our technology is just going, if we don't merge with our technology, in a few years, we will be like primates, robots and avatars will regard us as primates with a very rudimentary language, and so they will be able to do things that we are like if we were cockroaches or something like this, far impossible to do. So we have to merge. This is something that we wouldn't have a choice really, but to do this in the future. So that is another branch of electronics that is coming to interconnect. Elon Musk is working part of this.
Neuralink.
To interconnect, Neuralink, to interconnect our brain with our computers. That is not trivial, but it's definitely we need to do that. We need to do that because, yeah.
So how do you think we're actually going to solve the human aging problem? Can you maybe share details? And keep in mind, obviously, we haven't done this, so nobody knows for sure. But what's your best guess or what's your best thoughts on what we should be focusing on biorejuvenation technologies to actually, solve the human aging problem and keep us young?
Well, we have to, each biological systems, animals are very, very complex. We are really far from being able to understand the intimate mechanisms. And when I do my experiments with the Yamanaka genes, with viral vectors, my main goal is not really to understand the mechanism because this will take a lot of time. What I want to do is to just be able to use them in effective ways, to devise effective ways to use them for them to do what I want to do, which is rejuvenate. And in this regard, the Yamanaka genes can do it by mechanisms that I am completely aware of, I capable to understand, but they are doing that. So what I will focus my efforts in finding the way to harness their power and just neutralizing their harmful effects. So this is not easy, but this is the way. I think the Yamanaka genes have been one of the focus of attention and work in vivo work, because with cells, everything is done successfully. And this is something which is not minor. Rejuvenation has been fully achieved and successfully achieved in cells from all individuals, even from sentinel. So this is a major achievement. So this can be done. It's nothing hypothetical. It can be done. It has been done. If you have a laboratory where you have the capability to do cell reprogramming, you can rejuvenate cells, any kind of cells that you want. Okay, so this is done. But now we have to move to a next scenario, in vivo, which is the scenario that matters to us. And then things are much more complex and the goals are more challenging, the process of doing that. And so we need more people. This is why David Sinclair is doing so much good to the field, because it will give visibility not only to the public, but to the people that grant funding, because everything boils down to money in this world. So you need to get the interest, and there is something quite relevant in those studies. So David's study is really relevant and very important for rejuvenation. And there is another discussion I have had sometimes by email with David concerning aging. Is a disease or is a natural process? So I was telling David, well, if we are drinking just a cup of coffee, I will tell that I think it's not a disease, it's a typical process. But if in order to have the FDA consider aging as a disease and fund different development, I will happily lie and say that I believe that aging is a disease, no problem. But then when we come back to drink coffee together, I will say, David, I think that this is a natural process. Okay, so my ethics, it doesn't go doesn't go too high in this. But the main thing is to just get funding, because without funding you cannot do experiments. So you need minds to attract minds to the field, and money, like to construct the atom bomb. You need both things, but plus a war, of course. But well, the war is aging has 100% mortality. We are concerned by COVID. This is 100% mortality. So I am going to get it. You are going to get it. That is something, and so we should rebel. We are beginning to have the tools to do this, but we have to invest resources in this. And this is what people like Bill Falloon and others, Keith, Comito, are trying to really make people aware of that, and rich people especially. I hope they succeed.
Definitely the question of if aging is actually a disease, human aging, to me, it's just semantics. So you stated it so eloquently. If we have to lie and call it a disease to get the governments to fund research for it, then sure, we'll do that. That's why I like to say, let's solve the human aging problem. It's more, it kind of moves past that grammatical.
We will. It will take some time, but we will, because we are perseverant. And this is in our nature from the dawn of civilization. We want to live young and, if possible, forever. So unless you believe that it's a paradise and something, but I am a non-believer. I only believe in the human species.
Me too. Yes, I know what you're saying. So just an inside scoop here. I told you guys that I'm the host of Lifespan News and we do stories every Tuesday. So the rundown is produced by Sadir El Shok and Nicola Bagala, who are at, Leaf, employees of Leaf. I'm a volunteer. And so every Thursday morning I get the rundown from them. And there's we do 5 stories. So here's an exciting story that's coming up on Tuesday. The NIH, the National Institute of Health in America, opens call for funding. So the National Institute of Aging at the NIH is inviting applicants for a phase 1, 2A, and 2B clinical trials. The aim is to support efficacy and safety trials for such drugs or treatments. The announcement says a letter of intent is due in March, and the call will open in May. The NIA intends to commit $4.5 million USD in total cost for fiscal year 2020 to fund 4 to 6 awards. So this has never happened before. The NIH is now putting up $4.5 million essentially to fund aging research.
Well, researchers like David Sinclair with this kind of publications, high impact, are helping a lot to these things to happen. So I think that really those are as I say, the agents of change that will turn this idea of rejuvenating as a viable idea worth of being funded. Because so far, really small foundations, NIA is not too inclined to find a rejuvenation study that is not solving a specific problem. We're not curing Alzheimer's, but in fact, if we succeed, we will cure everything. If I prevent you or some person to get older, I am just dealing with the risk of Alzheimer disease development because all those are age-related diseases. So they will happen, whatever, cancer, whatever. So if you keep someone young or make it age more slowly, so if you slow down the epigenetic clock, the ticking of the clock, you are extending the lifespan and the quality of life of those persons. So this is, can you imagine something more important than this? Well, you are not curing a disease, but this is thinking in a small context. We have to give a relevant place to research on aging. And now I would say on rejuvenation, we have the tools now, the Yamanaka genes and other to rejuvenate, so we can start to develop the science of rejuvenation research. This was last century was not possible, but now it is possible, and the potential for that is really far-reaching. So I hope that these people that are so active in raising money can really convince the public that this is an important thing, because aging is happening to everyone. So I'm not so concerned. If I have a sister with mammary cancer, I am really more inclined to donate to Cancer Research Foundation. But since everybody aging is something normal, happen, I don't say I don't feel more compelled to really donate for aging research because it's happening to everything, to everybody. So except a few people that really rebel against this idea and try to fight it, like you probably, and like me. And if I die without really receiving the benefits of all those things, I will die happy that I contributed the human species that has given so much to me to well advance and get closer to that ultimate goal that we will reach. Definitely, we will reach.
Definitely. I believe so as well too. And, just to give some statistics to what Rudy was talking about there, on average, about 150,000 people die every single day on Earth. And about 110,000 of those people on average that die daily are dying from aging or age-related diseases, like heart disease being number one and cancer number two. So the idea is that if we can slow, stop, or even reverse aspects or the entire mechanism of human aging, then we can save all those people from the suffering and then keep them contributing in the world and just increasing our global economy, GDP, and increasing, how much things that we're improving in humanity. So that's the idea. And then we'll have to deal with overpopulation, but Elon Musk might, set up a colony on Mars and Earth. We'll deal. Those are good problems that we'll have to deal with.
I wouldn't be much concerned with that because it will take a long time to happen.
Right.
Because this will not happen overnight. So the other message to the public is that now in the 21st century, we have the tools to rejuvenate. Before we didn't. We could speak of nanotechnology, but that was kind of far away in the future. Now we have the tools. We can rejuvenate cells right now. This is nothing that is a theory. No, I can rejuvenate cells right now from old people, very old people, and from old animals. That is a miracle. So this can be achieved. Now we have the next step, which is a more difficult, more challenging one, rejuvenation in vivo. But there is nothing, no law of the universe that says, before, I remember in the first lecture when I began to study biochemistry, the physics professor said to us, well, you are young now, but unfortunately, because of the second law of thermodynamics, entropy, we will age. That was a very bad news when I was just beginning to study a career to defeat aging. So I have my disagreement with that professor. And well, I think that definitely the second law of entropy is not really condemning us to age. And we will fight aging as always we will fight aging because it's something that is in us. We will not want to perish, to disappear, because this what that we are this is only one chance in the whole universe that the molecules will come together the way they are to get another Brent or another Rudy. So this is why for the individual, death is a tragedy. Um, for the species, maybe not, but for the individual, it's a tragedy. And so we do our best to hang on that. And this is also why cryonics is something that is worthwhile trying. But this is another story.
Definitely. And one thing in the paper that you sent me a few weeks ago when we got in touch with each other that I read is, I believe you were talking about in there, well, to a certain sense and in a way, if you think about it, all species have already created biological immortality because we create copies of ourselves in reproduction. So I just had my first child. He's 14 months, about to be 15 months. And clearly there's a way that there's part of us, our germ cells, our reproductive cells that don't age, or at least they don't age at the same rate as our somatic cells, which is the majority of our cells that make up approximately the 40 trillion cells of our body. So clearly this is happening because my son was born at age 0 rather than at age 30, 4, which was the age I was when he was born. So the question is, what's going on here, and how can we take some of that, information that's in the germ cell that's biologically immortal, so to speak, and put that into our somatic cells? That's a
Well,
big question,
this is
right?
yeah, this is the only function I can imagine for the Yamanaka genes is just to make sure that in the first cells, the zygote, which will convey part of the age of the parents, even if they are young, because of the epigenome, the genes and the epigenome that they will carry part of that aging. Little small, little bit, but something. In the zygote, which is really the oocyte of the mother, those Yamanaka factors and others probably are present and set back the epigenetic clock of that zygote to 0. So in that way, we are born fresh with 0 age. Otherwise, if that mechanism was not present, we will accumulate little by little age. So we will be born not with age, age 0, but a little bit. But in the next generation, that will be increased a little bit more, and in the next generation, more and more. And this doesn't happen because of the Yamanaka genes that are resetting the clock, the age of the zygote to 0. Because it was always impressed me that babies were just always fresh and quite young, and there is no the, their parents were not passing any aging traits. And now, when the Yamanaka genes were discovered, it became clear to me why, because of them. But then, after they accomplished their mission, they got silent. So like Egyptian tomb. So and this is the genius of Gordon and Yamanaka that discovered it.
So it
So this
sounds to
is
me, with me asking that question about what should we be doing to try and solve the human aging problem if you had unlimited resources, I don't think you directly answered, but it sounds like Yamanaka factors is what you would be focusing on with all.
so definitely I will spend most of my resources uh, building a lab and of course establishing collaborations with other peoples working in the same area, trying to really push forward that, because if there are enough minds and enough money to push like the atom bomb, any subject, that progress will be made faster. And this is like a chain reaction. If there are more discoveries pointing the feasibility of that, more people will get interested, and so the field will grow big. Now it's a very small, but just you can count with perhaps the fingers of your two hands. Sinclair, Sebastiano, to a certain extent, Steve Horvath, and a few others. Ispizua Belmonte, Alejandro Campo, who is also trying to begin to do something, and a few others, but very, very small. That should be increased. This is why, again, I consider that what David Sinclair published was very, very important for the field, this emerging field. So I hope that David can keep producing these very nice papers.
Yeah, definitely. And there's, some popular YouTube channels that have done some work with David over the past couple of years that have, like over 1 million views. So you just didn't really see this stuff a few years ago. Venture capital has been rushing in to anti-aging, longevity, biorejuvenation space over the last 5 years or so. So these are all exciting things. The HEALS conference that Didier hosted that we mentioned from February 11th that you attended, I thought that was a great conference. It felt kind of like a ending age-related diseases conference that we had online in August, but all packed into 5 or 6 hours. So I guess we should be doing stuff like that more often and using technology. One thing that I'll just plug real quick is I just recently joined Clubhouse and I've been kind of playing around on it. If you guys haven't heard of it, it's a new app. It's only available for iPhone right now, but I'm trying to follow and connect with people who are interested in longevity. So jump on Clubhouse, follow me, I'll follow you, and we see if we can kind of have conversations. I think we just all have to network more together. I knew of your work, Rudy, but we hadn't even been in touch until a few weeks ago. So I think that the more we network and share information and figure out how we can help each other, the better, right?
Yeah, of course. Well, the pandemic has, because we are a faraway country, the pandemic has allowed me to travel more easily just online. So I interacted with more people during the pandemic. I am not meaning that the pandemic is good, it's bad, devastating. But some aspect of the pandemic have been, so to speak, beneficial to me because I interacted more with people because nobody now, most people cannot travel. So everything's organized online. And so I can attend and exchange views. So that is a good thing. But yeah, definitely we have to be interconnected. I think that this small uh, core of persons interested in reprogramming, rejuvenation-based reprogramming, we are in touch more or less because we are so few that we know each other more or less. So that's good. And your work is also very important because you are promoting really making the public aware of the. And this is the main point. Uh, Aging, whether disease or natural process, has 100% mortality. COVID has 5, and COVID is circumstantial. It will be overcome. Death and aging will not at the moment. Will keep there and will keep everybody given time.
Definitely.
So
And Rudy, another one of our stories that will be coming out in Lifespan News on Tuesday's episode is a new established ethics code for longevity journalism. So I've already read that. It's Leaf has produced that, and I think that it's really important that people read through that because in mainstream media, I see a lot of the mainstream media try to start reporting on longevity science, but this is so complex, and the way that media works nowadays, especially social media, it's all about clickbait titles and things like that. So the media almost always gets these longevity stories incorrect with regarding the science. In fact, even within that.
exaggerate, yeah, exaggerate, and giving sometimes uh, false hope that things can be just round the corner. And this is not it. Things are exciting, but they will take time, even those times that everything goes fast. So this is why Elixir, for example, when the Elixir preprint was published in bioarchives, the interest was enormous. In just a few days, um, over 100,000 PDF was just downloaded. So that had really, I realized through this, a very strong impact on the public. And now I am concerned, I don't want that this, I am concerned if a lot of expectations grow for Elixir because we have to confirm that it does what it does. Because if you give so much, so many and high expectations, and then it turns out to be that it's not, it was a mirage, something that is not what is seemed to be. Well, that is bad because then
in
it just
America, in America, we have this story that you're taught. I was taught it in kindergarten when I was 5 years old. The boy who cries wolf. Have you heard of that story before?
yeah,
So that's
yeah,
what
yeah.
it's like, we all have a tolerance. It's like, all right, sure, we're going to cure aging. We're going to solve the aging problem. Sure, sure, sure. You've been saying this forever. So yeah, we definitely want to be careful with that. And that's why, journalism and reporting the real science is so important, which is why we
We
really
are,
appreciate you being here and to share all this information with us.
well, medicine in general is pushing the limits of aging in the last century or so, really significantly far from when it was before. 200 years ago, life expectancy was, I don't know, 45. Now it's 75. Well, for women, more because they live longer than men. Females in general live longer. So medicine with technology and everything has really prolonged the life, but there is a limit. Medicine by itself will not prolong life indefinitely because we have certain fixed limits, which is just a dog is built to live no more than 15, 17 years in general, are about 3 years, and we maybe 120. But after that, we will have to make modifications in our software to just change that. And that could be done, definitely could be done. And it will be done. I don't know within my lifetime, but it's very, even if it is not, it doesn't happen on my lifetime, it's very exciting to watch, to be in the first row. And of course, be a participant doing something that we are doing in our lab.
Yeah, definitely. I agree. And I'm 35 right now. I'll be 36 next month in March 2021. And even if it doesn't happen in my lifetime, it's still the most exciting stuff that I'm obsessed with. I mean, I quit my job making a lot of money in Silicon Valley at Microsoft to focus full time on this because it was too exciting. And I felt like I had enough money to live on my own and manage my investments and just focus on this full time. So it's been, I mean, I could, read a lot of books and watch a lot of stuff on the internet, but I could only do so much working a full-time job for so many years. Now that I'm doing this full time, I'm able to really make connections with great people like you, interview the scientists, really understand what's going on at a deeper level. So I understand the problems that we have and the challenges to solve the human aging problem so much better. But I also understand the science, so much better as well too. So it's
Oh, yeah, that's good. Well, James Clement is a lawyer, but he was just captivated by the passion of doing aging, and how now he's fully involved in that, and he's also a cryonicist. So we share with James a few things. So this is another example. No, sometimes is something is like a bug that just bites you, and you cannot get away from that. So,
Yeah, definitely. James told me that it was his third year of law school where he got bit by the anti-aging bug and he wanted to even drop out of law school and focus, go get a PhD. And he said his wife at the time said, no, 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 no. She didn't like that. So he finished getting his degree and practiced law for a little bit before he made the switch into this, anti-aging biorejuvenation field. And yeah, James is a fantastic person. We're definitely rooting for what he's doing. On that note with James, I'm also friends with Kevin Parrott, the founder and CEO of Open Cures. And, I had a private Zoom call with Kevin a few days ago, and Kevin, loves James and what he's doing. But what Kevin said is that instead of working on doing human clinical trials on super centenarians like what James is doing, or even just centenarians, Kevin thought it would be more interesting to be doing human clinical trials on the children of super centenarians or centenarians. So people in their 70s or 80s before they're really, that old, to see the kind of results they're having from, at Better Humans, they're doing exosomes injections, NAD plus infusions. They're doing all sorts of types of treatments like that. So what are your, brief thoughts on that? Do you think that would be a better approach?
my my gut feeling, exosomes, I don't have much knowledge, but they don't hit me like being something like when I just read about the Yamanaka genes. I said that's it. Like when you saw the structure of DNA, you knew that the crystallography was well done, carefully done, rigorously done, so the molecule was. But when You had a look at the molecule, at the model of the molecule. You said, this feels right. This is a gut feeling because I didn't know anything. And when I saw the results from the Yamanaka genes, that hit me. This is the real thing. Exosome, telomeres, and so we could spend another hour discussing this. But Steve Horvath also has proofs and records that and experiments that show that those because just one word about telomeres. Telomeres shorten with replication, but there are cells, very important cells in our body, our brain cells, with few exceptions, and our muscle cells that are post-mitotic, that is, they not reproduce. Still, they age. So this is not. This is why this parish is just diversifying. I think that one of the things I have that things have to be looked in the genes, in the epigenome, and in the brain. I have a strong interaction with Steve and with Ken Rush, a close associate. We have spent some time together in Argentina discussing very interesting things, but he's a biochemist, has a view that where goes to the cell, the scenario, the scope of him is the cell, and I try to discuss with him that cells are not autonomous, they are regulated in a mammal by a hierarchical system, which is the brain, because the brain evolved to control. The blood didn't. So cannot be the blood the source of youth. You have to look at the mechanism to touch. If you're going to touch something, look into the brain, the hypothalamus probably, to discover some areas of relevance. This is my view. Of course, every component of the hierarchy has certain responsibilities and actions, like with the circadian clock. But you have a command center, you need a command center, which usually is in the brain. Of course, the brain receives input from everywhere, but that clock that may be modified a little bit, slow down, or speed it up, as a setup that cannot be modified. As I said, you can achieve that the dog live until 20, but never until 50, typically. Same thing with the rat. So our lifespan is fixed by a program probably, and this program probably is in the genes, of course, in the epigenome, but it's not just spread all of our body. No, everybody's not giving signal, like in a country, only the prime minister just order to the ministers, and then they give order. It's a network, hierarchy, a network. You're saying the Yamanaka genes are just at the top of the hierarchy. They issue orders, and then networks of other genes are activated because it's not happening with just our gene. We have no idea what those networks are, but this is the way one have to learn how to think like nature, like biology. So you have to think how biology would proceed in this way, because she has a pattern. So, try to imagine things in the way biology will do them, just trying to get closer to a right model. So this is my view of that, and I am trying to apply this as soon as as frequently as I can. The exosome, we have to see, because nobody in biology is an empirical science. So let's see what the experiments achieve, and then perhaps there is something very important there. I don't know. Just it's a first impression, but I may be wrong. I have been wrong many times, so could be wrong.
I can just share real briefly about exosomes. I interviewed the co-founder and CEO of a company called Kimera Labs back in October 2019 at RADFest, the Revolution Against Aging and Disease Festival. His name's Dr. Duncan Ross, and I've developed a relationship with him. And I actually know some medical doctors who I've also interviewed on my YouTube channel, Dr. Ed Park at Recharge Biomedical down in Orange County, California, who's been injecting Kimera Labs' exosomes into patients for a couple of years now. and including himself, Dr. Park and his mother, and Dr. Duncan Ross. He's a PhD. He's injected himself. Ed Park is a medical doctor, trained gynecologist, now been doing anti-aging for over a decade or so, practicing that. And the results are quite frankly phenomenal. And I don't think it's going to solve the human aging problem. This exosomes is not going to do that alone, in my opinion. But the therapeutic benefits, anecdotally and from some other more scientific measures, are pretty phenomenal. So Ed Park runs a blog where he kind of shares this information with people. So the blog's probably over 40 different testimonials at this point. And so the exosomes that Kimera Labs has created come from the placenta, one placenta of a healthy human birth. And so then they've scaled those up. And it's just the exosomes are just little microvesicles that came from the mesenchymal stem cells of that placenta. The MSCs, also called mesenchymal, or, Dr. Arnold Kaplan, who named them mesenchymal stem cells in the late 80s, calls them medicinal signaling cells, because what they really do is they send these signals to basically tell the other cells around them that the exosomes go into to basically behave younger. So that's kind of the real quick synopsis on my thoughts on exosomes. But you've seen all sorts of things, injecting them into knees. I had my neighbor a few months ago get them injected, the Kimera Labs injected into his wrists, where he'd been jackhammering as a day laborer for many years, and he wore down the cartilage there. And so he's gotten benefits from it anecdotally. So this was back in August, I think, when this happened. I think that's when it was, of 2020. And I just talked to him a few weeks ago, and he's doing better. So that's just one example.
Well, this is a um, very useful therapeutic tool has been discovered, and this doesn't take value away from that. If you discover a therapeutic tool, a compound that does good things, therapeutically speaking, that's good. That's very good. Doesn't mean that it's a rejuvenation uh, elixir, but anyway, discovering things that are therapeutically sound and useful, that's very good. I have nothing against that. So that there is no problem. So, but I am looking for a different thing. But this is my person concerning the mesenchymal stem cells, the adult cells. My own experience, I don't believe in flying saucers. Okay, friend. But if one day one lands on my backyard, I will begin to believe. Okay? This is what happened to me with this experiment. I didn't believe at all in mesenchymal stem cells, humans. I said they are from another species because I didn't know, I was ignorant about that. And my colleague, my friend, had really an insight, genial insight. Really, the merit is his, not mine. I just let him use my lab. And so I forgot about the rat because initially nothing happened to the rat. Yeah. But every other week he came to the lab with a syringe. My wife is a technician, injected the rat and so on. And I forgot all of it because the rat was not doing better or worse, was just okay. So it doesn't work. And I forgot. Then 2 years later, my students report to me, Rudy, look, this rat keeps living and the little mates are dying. So I begin to look at the rat again. Well, when I realized that something was happening because the rat kept living and living and living, was too late to try any. I only film and make a video of the rat and saw things. And the only thing that I could check, because rats, usually female rats, develop mammary tumors. I can provide you more information with this. It's very frequent. This rat developed no tumor, and usually female rat, for certain processes that happen related with estrogen, have their pituitary glands enlarged with age. So they may reach 50%, may become 50% heavier, or even 20%. So when you extract the pituitary of an old rat, say 25-month-old rat, female rat, you will find definitely an enlarged pituitary. Maybe normal, but enlarged. This rat, 44 months old, the pituitary was like the size of a pituitary from a 2-month-old rat. So that was the only thing that I could do because the rat died over during the night. And so I was regretted really very badly that I didn't have a more vision or insight to do some other studies on the rat because I didn't expect this result. So when you are have accepted about a result, and the result happens, that makes the result more objective because I was expecting the opposite, you know. And now I definitely believe that mesenchymal themselves do things important, do not rejuvenate, but stabilize homeostasis. And making the treatment is not invasive, just injection. Well, after that publication, some persons in Argentina asked my colleague, who is an MD, to be treated with those cells, but he couldn't do it because he would be experimenting with humans. That couldn't be done. But I would, for one, would be ready to just inject those cells because I saw it with my own eyes. It's not just something that I read. So I can with certainty tell you that mesenchymal stem cells do something if you treat people or animals when they are young adults. They will regularly. They will probably live a longer life. The rat aged, I mean, but they aged gracefully. So they kept their mobility, but looked like old. That definitely it was not rejuvenating, nothing like it, but with a good quality of life. So this is my greatest achievement. Was just a case report, but this was the more successful serendipity had a role here. Results that they had. But anyway, I cannot do these experiments. I wanted to publish this or just to encourage other people with more resources than me to try to do this. But I am now focused on rejuvenation, and I will just keep doing that.
Awesome. And we want you to continue the fabulous work that you're doing. And I really appreciate you taking the time to do this interview, Rudy. And we're actually 15 minutes over from the time that we agreed upon. So
Oh, okay.
I want to,
Let me make just a last comment.
yes,
In the
please do.
interview with Richard, his last question was, "Are you planning on retirement?" And I said, "Well, when I started science and doing research, I just purchased a one-way ticket, so I'm not coming back. I will just the only stop will be when my health just declines, and so my brain stops working. That will be my retirement. But I am not planning to go back home and watch TV or something. I couldn't. I just would die very soon. So this keeps me alive doing what I am doing and doing this thing. So I have been like you. I have had the privilege of being able to work on what I like to work, so to do what I want to do. That not many people can say that, and we can do that. And this is I am very grateful for this opportunity that life and my parents gave to us. And well, I will keep doing what I do as long as I can.
That's beautiful. That's a beautiful way to end it, Rudy. And I appreciate you sharing everything that you have with our audience. It's been a pleasure to get to know you and communicate with you. And I know that I've made a friend for life, and you're very, very valuable member of this community. And I think in the short term, we're excited to see, your young plasma transfusion and your rats experiment that's going on right now to see how this will play out over the next year.
Yeah, well, at the end of this year, we will have some results, definitely, good or bad, but some results. And the more important thing is that Harold will have results too.
Exactly. It's very important. So, Rudy, thank you so much. It's been quite a pleasure. And yeah,
Thank you, Brent.
any final words?
Well, I received a message from LinkedIn saying that you wanted to communicate with me, but that you meant but type in a video conference, something like this, just communicate by messages via LinkedIn, because I received just today a message from you from LinkedIn. but I didn't understand if you wanted to communicate like this or just exchange messages. But I rarely use LinkedIn. I just use email or WhatsApp.
Yeah, we
So
originally connected on LinkedIn. I reach out to a lot of scientists and entrepreneurs in longevity on LinkedIn, and then we connected via email after that too. So email works, but I'm glad we were able to connect, and I'm glad we're able to do this interview, and hopefully the audience have found it valuable and learned a few things. So I thank you so
hope
much.
so. And you can distribute my email freely because I will be glad to respond to questions from the audience.
Perfect. Yeah, that's a very important thing there that I normally ask Rudy. So I check the description below, everybody. Check the pinned comment in the description. We'll have a link to Rudy's email. So feel free to get in touch with him, ask some questions. He's happy to respond back and help us get closer to solving the human aging problem.
Public paid for my education, my research. I think they are entitled to know what I am doing.
That's a beautiful perspective, Rudy. And that's how I feel too. That's why I'm doing this YouTube channel. We got to get this information out to more people and get more people excited about it, bring in more funding. It's happening. We've talked about it during this interview with the NIH, now funding the NIA $4.5 million for fiscal year 2022. Things are happening. We just
Yeah,
want them to happen
yeah,
fast.
will happen. Yeah, exactly. Well, let's hope that it will accelerate because I am running out of time.
Yeah, exactly. And Harold's 76. Harold Katcher
Right,
76. He's no spring
right.
chicken. You're 69. So that's one thing I'll talk to Harold about. I don't want to lose you guys.
Well,
This is my longevity spaceship here that my other
uh,
co-founder of my longevity plan company is Vladimir Trofanov, and the other one is Anton. He's in Ukraine, and Vladimir is in Russia. Vladimir's wife, Anya, is a beautiful artist. She made this for me. This is our longevity spaceship, and I want this to be real someday.
oh,
I want to be on this spaceship with you one day, Rudy. So that's
okay.
the goal. Yeah.
By the way, do you know Yuri the giant? The giant is difficult to pronounce his name. He's a Russian Canadian,
Yuri
very
Deegan.
young.
Yuri
Yes,
Deegan.
exactly.
Yes, I interviewed Yuri on this YouTube channel, my personal BrentNally YouTube channel at RadFest in early October
Yeah.
2019.
He's quite well informed, a very young man, and our brains work like twin brains concerning aging and the mechanisms. So I very much enjoyed discussing things with Yuri. And if you know him, okay, because he's extremely well informed about everything that goes on aging, on the ageism research and business.
Yes, I completely agree. Yuri was great. I'll put a link in the description below here at the timestamp of when I interviewed Yuri in October 2019, and he's fantastic. I completely agree. He's very intelligent and very driven. So I was honored to
Yeah,
interview Yuri in 2019.
yeah, yeah.
All
Okay.
right. Well, I think
Thank,
now's a good time to end it, Rudy.
yeah, thank you for the invitation.